This video was requested as an educational resource for the North Carolina Business Committee for Education, or NCBCE. The goal is to share knowledge about science that's related to the COVID-19 pandemic. My name is Dr. Jared Heffron, and I serve as a microbiologist for Novozymes. Personally, I have a background in infectious diseases from my career before Novozymes, and I have experience in teaching that I've been using nearly every day for the past 20 years. Novozymes does not work in this space. However, we want students to get an opportunity to learn from my expertise and build a better understanding of the basics around what is indeed a truly unprecedented situation. Hello, my name is Dr. Jared Heffron, and today I'm going to talk with you about viruses. I'm a microbiologist, which means that I'm a scientist that specializes in studying microscopic organisms. And viruses are one of those. So the word microscopic means something is so small that you can't see it with your eyes. You have to use a special tool in order to see them, like a microscope. And what's really important to realize is that we live in a microscopic world. We live in a world filled with microbes. They're everywhere. They're on us. They're on our skin. They're in our hair, they're in our mouths, they're in our stomachs, they're in our intestines, they're in the water outside, they're in the water we drink, they're in the ground that we walk on, they're in the sandboxes that we play in, they're on our pets, whether you have a dog, a cat, a bird, a lizard, a fish. They have their own microbes on and inside of them. They're on plants that we plant in the garden, they're on the plants that are out in the woods. Almost all the microbes that exist in our world are harmless. They don't make us sick. They're actually really important in keeping us alive. In fact, if it wasn't for the microbes on our planet, we would have no oxygen in our atmosphere at all. So, thanks to them, we all have a place to live. But sometimes we do get sick. Sometimes we wake up and we have a sore throat, or we have a fever, we're just not feeling well. That means that there's some microbe inside of us that's making us sick. That group of microbes that has the potential to make us sick, we call pathogens. We say that they are pathogenic. Most bacteria are not pathogens, but some are. Most fungi are not pathogens, but some are. And most protozoans are not pathogens, but some are. Now, those are all different types of microbes. There's another type, of course, viruses. The thing that makes them different, one of the things that makes them different, is the fact that all viruses are pathogens. And that's because of their lifestyle. In order for them to make copies of themselves, in order for them to reproduce, essentially, they must infect something else. And because of that, because they can't live and grow on their own, they're always pathogenic. They're always hurting another cell in order to make more of themselves. So what is a virus? We've already started talking about it. It is a specific kind of microbe. It's really itty bitty 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 tiny, and it cannot survive or reproduce on its own. It has to infect another living cell. Any cell that's infected with a virus, whether it is a human cell or a dog cell or a cat cell or a plant cell or a fungal cell or a bacterial cell, is called a host. So virus infects another cell, the cell it infects is called a host cell, no matter what. And you heard me right too. The virus can infect a bacteria. A virus can infect a fungi. That means it can infect another microscopic organism. And we're going to see this in the next slide, but that's because viruses are so small. They're even more microscopic than things that we consider microscopic. They're 1,000 times smaller than our cells, 100 times smaller than a bacterial cell. In fact, most viruses on this planet infect bacteria. They're not interested in our cells whatsoever because there's just so many bacteria out there. So it's incredible. There's an entire world. There's a war going on our planet at all times between viruses and bacteria everywhere you can possibly imagine, especially in the oceans, and we're completely unaware of it in most instances. I think that's incredible. So here's a way to look at that size difference between all these different microscopic things. Human cells are microscopic, and here we can represent it as this nice big squishy beach ball. Bacteria cells are that much more microscopic than a human cell, and here it is as a tennis ball in size. You could put a whole bunch of those tennis balls inside the beach ball, right? And then a virus particle is even that much more smaller than the bacterial cell. So now it's just a speck, it's just a dot. There's not even a ball that's small enough to represent a virus particle in this analogy. So just hundreds of those specks could fit inside the tennis ball, and thousands, if not millions of those specks could fit inside a human cell. So all these things are microscopic, but there's different levels of microscopic. We get smaller, we get smaller, and we get smaller. So how do we see viruses? Fortunately, we've made a lot of different tools over the last years, decades, centuries that help us see, well, microbes in general, but also viruses. Now, the first thing to keep in mind is that a light microscope, like this guy in the upper right-hand corner, will not help you see a virus. It looks really neat and complicated. It's great for seeing bacteria and fungus and our own cells in our body, but it won't do a virus. Instead, you need a much more complicated microscope, which is down here in the bottom right, called an electron microscope. Much more expensive, much more involved, and it actually doesn't give you that amazingly good of a picture, but because they're so tiny and small, that's what it takes to get a grainy black and white image of a virus, which I'll have one for you guys a little bit later to see. Most of the time, we use molecular tools in order to measure and see and track viruses. Molecular tools, that's using biochemistry and genetics, molecular biology, in order to help you indirectly see what's going on, where the viruses are and what they're doing. It's very complicated. It's outside of the scope of this presentation, but it's an exciting field. Perhaps we'll get to talk about it at a later time. Main thing to remember though, viruses are incredibly small. You're going to use the metric system. They're about 1/10 of a micron in size, if not even smaller, 1/10 of a micron, 1/10 of a micrometer. Bacteria, they're a whole micron in size. They are giants in the world of biology. They're enormous. It's hard to imagine something can be that simple yet that large, right? And then there's us, the animal cells that make up our bodies, 10 to 30 microns in size. We are colossal. We are titans in this world, and we forget about it every single day. I love it. This stuff is so cool. So what do viruses look like? Well, if I was to draw a cartoon, which I did, they'd look kind of like this. They are simple, right? Kind of looks like a circle with some neat little extra parts attached. In the middle, you have their genes, which are made up of, it's called nucleic acids, but we just call it a genome. It's a collection of genes, like DNA, or even more complicated version called RNA. And this is going to encode all the genes that help make up the virus. We'll see how that works a little bit later. Just like we have genes in our body that gives us different colored hair and different colored eyes, different colored skin, and it helps make us whether we're tall or short. They have the same kinds of things going on here. All right, so then their genes are really important, and it's protected by a layer of protein called a capsid. And you can see this series of proteins making the shell, this capsid around it. Some viruses have an extra layer on the outside of them called an envelope, which is made out of a membrane, kind of like the membrane in our own, so that surrounds our own cells. So again, this is very simple compared to other cellular organisms. If we got into the architecture of an animal cell, or a bacterial cell, or a fungi cell, there's going to be a lot more going on with them. These guys are essentially very simple. Again, they don't need to eat, they don't need to drink, they don't need to grow, they stay this size, they just hang out like this until they infect a cell, and then we'll see what happens in just a little bit. So here's another picture of it. A little bit more complicated, 3 dimensions, but you can see those same major structures you just talked about. Genome inside, protein capsid in the green color, and then a capsule, an envelope, capsule is a bacterial term, envelope for the virus around it made out of membrane. And then here now in the lower left, we've got an electron microscope image of a virus. So that's an actual picture on the left, not a cartoon of a virus. And this is an influenza virus that we're looking at. And so you can see my cartoon on the right of the influenza virus, and you can see an actual picture using electron microscope. And so, right, the electron microscope costs like millions of dollars, and that's the best picture we can get of these viruses because they are so small. Real grainy, a little out of focus, but that's what we can do right now. Maybe in a couple more years, one of you can grow up and invent a new tool that's able to give us better pictures. So here's extra credit information about what viruses look like. Just like every other thing you can imagine in biology, there is different shapes and sizes to them. We can have polyhedral or icosahedral viruses. So here's this 20-sided virus right here. There's little different shapes to it. I think they're very elegant looking. You can have a spherical one, of course, like influenza, which we were just looking at. Here's a different way of looking at it. There's helical ones that also kind of look like a rod, tobacco mosaic virus. And then look at this complicated guy. So we just call it a complex complex virus. It looks kind of like a spider, right? Well, actually, even though it looks fearsome and nasty, this is one of those viruses that actually doesn't do anything to us whatsoever. This is the one that hunts down and infects only bacteria. That's why it's called a bacteriophage. So yeah, these guys, as bad as they look, for us, they're harmless. For another bacteria, that would be a very scary sight indeed. And so yeah, these guys are all, this one can infect humans, and this one can infect humans. This one infects a plant. This one infects bacteria. Again, different shapes and different jobs. So now we know what viruses look like. What do they do? I've already mentioned this a little bit. Now let's talk about it in more detail. They want to transmit themselves to other cells, transmit themselves to other hosts, infect as many hosts as possible, because while they're inside the host, they're able to make more copies of themselves. So they attach to that host cell, they inject their genome into that host cell, they hijack the cellular machinery of the host cell, make copies of itself, and once there are enough copies of that virus made inside the host, they pop out and spread to the other cells that are usually nearby. They transmit themselves to other cells and repeat the process to make more and more and more of themselves. Over the course of this whole process, as you can imagine, it definitely hurts the cell, the host cell. And most of the time, often, it will also kill that host cell. So let's look at that in just a bit more detail. So let's check out a cartoon of an infection. And you start off with a healthy cell. Okay? It says host cell. It's not really a host just yet. There's no virus here. It's a happy animal cell. Remember that big squishy beach ball we saw earlier? That's what this guy is. So this is an animal cell. Let's say it's a cell from my lung. Okay? So a whole bunch of these make up all the lung tissue in my body. This is just one of them. It has a bunch of structures inside of it. Big circles, little membrane structures here, and mitochondria. I don't need to worry about what all these things are. They're called organelles. Just like you have organs make up your body, we have organelles that make up individual cells. They all have special jobs. Anyway, they're all doing their jobs. Cell's happy. He's just perfectly growing and eating and getting bigger and taking care of the tissue that he is. My lungs, right? Making me have nice, happy, healthy lungs. And now I've just suddenly breathed in an aerosol that contained a virus, unfortunately, and it got into my lungs. And so this virus is now close to my lung cells, my animal cell here, and it might come in contact with my cell. What's going to happen? Well, if it's the kind of virus that can infect a lung cell, then it will come in contact with the lung cell and bind to it. You can see those little proteins on the outside edge that are sticking out from the virus. They stick and adhere to the surface of my lung cell. And right after it sticks on there, the first thing it does is it takes its genome. Remember those genes that are inside the capsid? Here I'm showing them squirting through and being injected into my lung cell. So the virus binds, inserts its genome. And then, oh my gosh, what happened here? That was so quick. Yeah, I skipped a couple of steps. Basically, all of a sudden, you see that we've got a bunch of copies of that genome now scattered all over my lung cell. Well, it does take a little bit of time for that to happen, but essentially, like I said before, the virus genome hijacks the machinery inside the host cell and starts off by making a bunch of copies of itself. And so now we've got copies of viral genomes scattered all over my lung cell, all inside of it, which of course now is a host cell, right? Because it's infected. And so it's got lots and lots and lots and lots of copies of itself. And now, there's even more stuff has happened. We've skipped ahead in time. And after all those genome copies are made, it starts making copies of the capsid, that protein that can surround the genome. And again, the virus is using the host cell machinery in order to do this. And also, you see, the host cell doesn't look like that big, squishy beach ball anymore. It's kind of getting a little wavy here on the edges, not looking so good, starting to feel a little sick. And it's because the virus is using so much of the host's energy and proteins and, again, like I like to call it, cellular machinery, that the host cell doesn't work the way it's supposed to anymore. It's sick. It's infected. It's getting hurt. And then, after enough time and enough viruses have built up inside the host cell, it bursts. It bursts, and the host cell, of course, dies. From this point, now all the viruses are free out here in the environment. And in my lung tissue, you're going to have another animal cell right next to this animal cell. And so these viral particles can immediately interact and infect the neighboring cells and continue this process. You also see on the outer edges of the virus, it has those proteins sticking out on the surface of it again, right? There's the capsid encasing the genome here in the middle sections, and this extra layer, the envelope, it's a membrane. It's actually very similar to the membrane on the animal cell, because as it bursts out, it encases itself in some of the membrane and proteins on the edges of that animal cell. It's like a horror movie. It's coated itself in the skin of its victims, and that helps hide it from the other animal cells too. They don't realize that it's a bad cell because it looks on the edge just like another animal cell, which they've always next to and they're always touching at all other times. So they don't know until it's too late that this viral particle isn't really a friend, it's an enemy. And then this process is able to repeat again and again and again. And you can see how just one host cell creates potentially hundreds, if not thousands, if not even maybe millions of copies of the virus. So here's extra credit knowledge about what viruses do. Again, it was just scary, right, what I just showed, because it seems like one virus is just going to infect one cell and just spread everywhere and destroy everything it possibly touches. In truth, most viruses are very specific for which kinds of cells that they infect, certain types, only with certain proteins on the edges of them. Remember that little stick that was on the outside of the virus? Well, that has to be very specific to a part of the host cell in order to make it bind. So not everything gets sick all at once. Also, I said that the viruses hijack the cellular machinery of the host cell, which is true, but in some instances too, they hijack the machinery and make more copies of their own proteins as well, their own polymerases or an enzyme to make copies of their genome. So it can be a little bit of a fusion between virus-derived machinery and cellular-derived machinery. Right, this depletes the energy and resources of the cell. When it breaks free of the host cell, the fancy word for it breaking is called lysis, or the cell lyses. So the process is called lysis, and if you see it happen, you say, oh, that cell just lysed. And then your host cell membranes, again, those are used to surround the virus and make the envelope the skin of their victims. So here's my advanced illustration of what's happening during the infection. Exact same process we just went through before. We're just going to do it now. More details. So we're zoomed in even more than we were before. So here is our animal cell in the bottom half of the screen. It's so big it can't even fit it on the entire screen. So here's that membrane, and here's the inside of the animal cell. The cytoplasm is the jelly that's inside where all the organelles float around. And then here's one of the organelles, the nucleus, which houses the DNA, the genome for the animal cell. All right, we've left out all the other stuff. And then this is a lung. We'll stick with that. And we've just breathed in a viral particle. and now it's floating around outside. And the protein on the outer edge of that viral particle is a match for a receptor, another protein on the outermost edge of the animal cell. So those two match to one another. Remember I said it had to be specific? So here we go, and we see binding happens. Virus binds to the receptor using its own protein to the animal cell. Then remember I said that this outer envelope, right, is a membrane and has proteins from another animal cell that this virus had infected previously. So the animal cell thinks, oh, well, that's just one of my friends, one of my neighbors right there next to me, and doesn't immediately worry about it. It actually says, hey, let's get in together. Let's get close and touch each other. And the virus says, all right. And it then creates fusion between its membrane out here on this envelope and then the cell membrane and sticks itself right there inside the animal cell. At this point in time, I would say that this animal cell is now infected. The viral particle is inside the animal cell. And then immediately, before the animal cell has a chance to do anything else, it secretes, it sticks, injects, whatever word we would like to use, puts its genome inside the animal cell. Fusion is this fancy word where the viral particle and the membrane of the animal cell merge together. So now that genome, the nucleic acid of the virus, is floating around in the cytoplasm. In this example, it's RNA and not DNA. There's different viruses. There's DNA viruses and there's RNA viruses. This one just happens to have RNA. So now the viral RNA is free inside the animal cell, inside the host, and it's going to get transcribed, reverse transcription is going to turn it into viral DNA. So the RNA wants to become DNA because it's a little bit more stable, it'll be easier for it to make copies of itself. Remember I said sometimes there's enzymes that the virus uses, not always necessarily the cell machinery. Well, usually this process is carried out by a protein that's also coming from the virus that makes this switch from RNA into DNA. So now it's ready to make copies of its genome. So the DNA manages to find its way inside the nucleus, which is where the normal host cell DNA is, and it inserts itself into the host cell genome. Integration, it integrates itself in there. From this point, the host cell machinery pretty much takes care of everything else because normally it's going to be transcribing the genes in its own genome. Now the viral genes are in its genome, and so it's able to transcribe them back into RNA, back into what looks more like normal animal cell RNA. And from there, you're going to be able to make more proteins. Of course, those proteins are viral proteins. And so we can see the capsid now coming out of the nucleus. And we also see some of that RNA also coming out because remember, that's a genomic material that's normally inside the virus. So it wants to make more copies of that. So it has those to stick inside the capsid. It makes a whole bunch more of the capsid. Here I'm just showing one, but like in the previous cartoon, right, it could be hundreds, it could be thousands. That whole process of making the protein, the capsid, that's translation. Again, done entirely by the host cell machinery. And then all the parts are there and they assemble into the core structure of the virus. The genome, the RNA genome inside the protein capsid. It looks almost complete. So now it's time for the virus to leave its host cell. And the process of it getting out here is called budding. So an invagination in the membrane occurs of the host cell. The viral particle pushes itself inside. You can see there's more assembly of some extra proteins. There's that membrane getting around it. Remember the skin of its victims, so it's encasing itself in that. And eventually this pinches off and is released. And we get a complete virus coming out of its host cell. Now you can imagine if you've got hundreds of thousands of these viruses coming out of the cell at about the same time, this is why the host cell lyses. If all of these things bud and release within a short window of time, it just punches too many holes all at once in the host cell for it to manage, and that's where you get that lysis. So it's possible for the cells to just slowly leak the virus. It's also possible for them to all of a sudden erupt at once. And there's a little bit of both things happening in most infections at all times. So now for some extra credit. Well, all that process we just described is in truth really only one way that an infection might occur with a virus. And we call it specifically the lytic cycle. Again, that word lysis, lytic, meaning that the host cell bursts, right? And so it's kind of an aggressive type of infection. We also could say it leads to acute disease. So there's differences between the diseases. The acute are where you suddenly get all the symptoms all at once and it's very aggressive and it's happening now. And this is what most people think of when they're sick, when they have a lot of strong symptoms and they're really inconvenienced by it and they can feel it. Okay, an acute disease. And then here is another figure showing that happening, all the stuff we just discussed, where the virus infects a cell, and makes copies of itself, and makes proteins, and eventually bursts out. Lytic cycle. But there's another cycle that a lot of viruses can do, where it doesn't necessarily do this right off the start, and it can be latent, go into latency, or also we call it lysogeny. And instead, the virus goes dormant for a period of time in a host and then can become lytic later. Because remember that integration step where the viral genome gets inside the host genome? Well, actually, sometimes it just stays there. It just stays there, and the host cell just maintains that bit of DNA, not really realizing it's viral. And the host cell, of course, is going to continue to eat and grow because it's a living cell and makes copies of itself and makes copies of the viral genome at the exact same time. In a way, this is the virus is being successful here. It just wants to make copies of itself and transmit itself. So here it's just getting spread to more and more cells and in more and more places without having to be aggressive at all. But at some point in time later on in these cells' lifespan, for whatever reason, the virus DNA pops out and it goes back into this lytic cycle and then can become aggressive and cause lysis once more. This process here, lysogeny, depending on the host and depending on the virus, can actually be years and years and years. The example I really always like is chickenpox and shingles. Now, for the kids of this era, chickenpox isn't really anything to concern ourselves with, or they don't concern themselves with it, because they can get a vaccine to prevent the virus from ever really infecting them in the first place. But for my generation and those born near my generation in the 80s, and then of course those who are even older, chickenpox was just an expectation. You're young, you're a child, you get chickenpox because it's highly contagious and it's all over the place. And true that I got chickenpox twice as a child. And it infects you and you have a lytic infection, and that's when you get all the sores and blisters on your skin and everything, and eventually you get through all that. But what happens is the DNA from that virus is still hidden out inside your cells, and you grow up and you grow for the next 10, 20, 30, 40, 50, 60, let's live 80 years and make many copies of the virus inside all of your other healthy cells. And then you get old and your immune system gets weak and your body gets a little bit more strained, and suddenly the virus says, oh, you know, it's easy for me to create a more aggressive infection now all of a sudden. And it pops out and goes lytic again. And so what'll happen is sometimes people, when their immune systems gets weakened, dampened, they get cancer or they just get older, like I said earlier, then you'll get this lytic infection again. And then in older people, it's not called chickenpox, it's called shingles. It has a little bit different set of symptoms, but it's still a very uncomfortable, actually rather painful in the shingles form of a lytic cycle of an entire infection route that included first a lytic stage as a child, then a lysogenic stage through most of adulthood, and then another lytic stage at the end of adulthood in elderly age. So that concludes my introduction to viruses. Yes, I know, introduction. It actually goes much deeper than just this. But that's more time, more knowledge. If you're interested, there you can build a whole career out of studying viruses, virology. Or if you want to know more about all microbes, you call it microbiology and become a microbiologist, like myself. Or you can just have a healthy appreciation for this, learn about it, read about it, YouTube, Wikipedia, books, and just be knowledgeable about it and be a very smart person and have a good time studying other things as well too. All those are fine. So, like I said, I'm a microbiologist. I work for this company, Novozymes, which is a biotechnology company where we use microbiology. Specifically, I like to think more about the bacteria in order to use them to make things that we can use every single day. So I don't worry so much about diseases. I don't focus on that. Instead, I'll be using microbes to help make cleaning solutions, help make additives that go into our food, help make food for other animals like chickens and pigs, and help grow things that will become food for us one day like corn and soybeans, and use the power of the bacteria in order to make those things work faster, work better, and more available for us. So it's a whole world. I think it's amazingly exciting, and I enjoyed sharing some of it with you today.